Welcome everybody here to Iowa State University Extension Dairy Team Monthly Webinar Series. We continue today talking about the Asian Longhorned Ticks and how they entered Iowa. Our moderator for the day will be Dr. Philip Jordan. He's our Extension Dairy Veterinarian. Dr. Jordan, podium is yours to kick the program off.
All right. So I assume everyone on this that's joined us today knows that we have now found Asian Longhorn Tick and Theileria in Iowa, which is why we're having this meeting. This is probably it's a bigger deal for the bovine industry, especially cow-calf operations. But we do have some people that raise their replacement heifers on pasture, and there's some pasture dairies. So we thought this would be also of interest to the dairy producers in Iowa. So today our speakers are going to be Matt Brewer, who is teaches parasitology here at Iowa State, Grant Duell, who's my counterpart on the beef side. I'm one of the dairy extension veterinarians, and he's the beef extension veterinarian. And then Chelsea Hamilton is going to be joining us for a question and answer if we have questions about the regulatory side. She works for both IDOLs and teaches the regulatory stuff here at Iowa State. So today we're going to start with Dr. Duell. So Grant, the podium's yours. If you want to introduce yourselves a little bit more, you can, or if you just want to start.
You bet. Can you see my slide all right, Phil?
It looks good. Yep.
Okay. All right. So yeah, I'm Grant Duell, Extension Beef Veterinarian here at Iowa State University. And I'm by no means a entomologist or a parasitologist, so we'll kind of cover some of the basics of both the Asian Longhorn Tick and Theileria, which is the protozoal parasite that it also can carry. So, and then Chelsea, if you have anything to add as I go through, just interrupt me and we'll go from there. So, so as Dr. Jard mentioned, we've identified the Asian Longhorn Tick in Iowa this early summer. It was, which is expected. If you look at the distribution of the Asian Longhorn Tick, it was first identified in 2017, I believe, in New Jersey, Virginia type area, and then has slowly been spreading eastward from there. It as expected. It's somewhat surprising that it took it this long to get here. It showed up in Missouri 4 years ago, and if I would have been betting, I would have said it would have showed up in Iowa in 2022 or 2023. So I guess in somewhat we're probably lucky that it's lasted this long. But I don't think that it just showed up this spring. In fact, if we look at the amount of infestations that we saw in some of these cattle in southeastern Iowa in early June, probably pretty indicative that tick had been there for a while and just hadn't really reached the population levels that it was easily identified. And in this spring, we kind of went over that mark where we were seeing thousands and thousands of ticks on individual animals. And so, but in order to get that level, they really had to have been there for at least a year, probably two. So again, it's not unexpected, and it's something that we'll deal with. Also, Michigan showed up positive this spring. From what I can tell, that was not the case in Iowa was a animal that was, or a group of animals that we had some disease and death loss in, and the tick was identified. From what I can understand, in the Michigan, it was a tick surveillance program where they found the tick on a tick surveillance going through a natural area. And as far as I know, they haven't actually found it on cattle yet. So that's more of a one-off for Michigan, I think, compared to what we typically see. So again, I mentioned the severe infestations, and so this is a picture of one of those animals from Van Buren County down in southeastern Iowa. You can see the massive number of ticks here on the tailhead. That's one of the favorite spots. The ear is also common, and then down along the perineal area and into the flank. So here you can see on this bull the number of ticks down here in this tail, down in the perineal and area, etc. So if we had, there's a picture, I don't throw it in here, but there's a picture of their flank as well, and there's extensive ticks there. So again, we're seeing not just a few ticks or 20 ticks, we're seeing thousands of ticks on an individual animal, and a lot of that infestation that's pretty common with the Asian longhorn tick. So what makes the Asian longhorn ticks somewhat different is that it is has the potential for asexual reproduction, and in probably it's not a chance for that. That seems to be its major role of reproducing, and that that tends to be the pretty much standard, and the chances where it actually mates with a male is probably pretty rare, and that's one reason why we see these massive numbers is we have a tick that is able to reproduce on its own. Otherwise, we'd expect you know, it would take a male and a female being on the same animal in order to mate and then produce those eggs. So, but it is kind of standard 3-host tick, where we have the larval stage, which is pretty small, that can basically take a blood meal off of any host that it chooses. It then falls to the ground and goes through a stage, becomes the nymph stage, where again takes a feeding from a host, then transforms into that adult female, and then when that adult female has that blood meal, she will then fall off and then start laying eggs, and will lay several thousand eggs. Again, in most ticks, this is where the mating with a male occurs. Again, in this tick, that doesn't necessarily have to happen. And so, again, this tick basically can get a blood meal off any host. It tends to prefer the ungulates, you know, cattle, deer, those types of animals. But it's not. It will take a blood meal off anything. You know, birds are a common way that it gets moved around, in that it can take a blood meal off a bird, and then the bird can travel many miles before that tick falls off. And so that's one way it gets around. I think probably here in the Midwest, we probably see a lot of movement due to the white-tailed deer, where they are moving around and dropping those off. But again, it can those probably are main methods of that getting moved around. But because it only takes one female tick to expand, you know, if you take a animal from southeastern Iowa up to northwestern Iowa, and it's got a tick on it, then we've now got ticks in northwestern Iowa. So it's going to be relatively easy for that why this tick is so proliferative. Generally, it takes about 6 months for this stage to go through. That's if it happened consecutively. But we typically see, because these ticks in any stage can go into hibernation or diapause over the winter, then it's typically probably more like a year that this life cycle actually occurs. And so that's why I say it, in order to have the number of ticks that we had in southeastern Iowa, that didn't happen this spring, that happened last year or the year before that started, in order to get that many ticks through that stage. And so kind of the typical cycle that is reported is that the adult is feeding in the spring. It lays those eggs in the late spring. We then have the larvae in the summertime. they're going to drop off, have a blood meal and drop off, and then overwinter, typically, and then molt in the spring into that nymph, and then we go through that into adult again. And so that's kind of the typical way that that's described. Again, they're like most ticks, they are fairly winter hardy. We probably get, I've seen estimates of 30 to 50% reductions in ticks during over that winter time. But when we're dealing with a million ticks, 50% reduction still leaves us 500,000 ticks, which is enough to get that started again. So as we go further north, that's going to be a little bit more more likely to happen. So in this southern Iowa, where the winters are somewhat a little more milder compared to our northern Iowa, we expect that tick to be pretty hardy in that area. It's going to be a little harder for it to move into northern Iowa, but it's got the potential that it can do that as well. So I'm not, but it's probably going to take it a little while unless we start moving cattle around. It's going to take it a little while to get to northern Iowa, probably. This is a forecast. Some entomologists looked at what the potential range was for the Asian longhorn tick. And you can see that here in southern Iowa, and then kind of up that Mississippi River Valley is a suitable area. Again, this forecast says that northern and western Iowa is probably less suitable. I'm not sure how true that is. I mean, we've got plenty of dog ticks and other deer ticks, etc., in the rest of Iowa, and I'm not sure why the Asian longhorn tick isn't as environmentally hardy as those ticks are. So I'm not sure that this range is really going to be accurate. I don't see why it won't spread into other parts of Iowa. Definitely, where we have southern Iowa and that Mississippi River valleys definitely has a nice environment. It's a little more humid. There's more brush, grassed areas, which the tick prefers. But again, we have plenty of ticks in central Iowa where mostly we have corn and soybeans, which isn't tick habitat, but there's plenty of other brush areas, etc., that they can survive in. So I don't really believe this is going to be the long range, but it's probably the most suitable for it. But it's not going to stop this tick, I don't believe. So that's a little bit about the tick that we're dealing with. The other problem that we've ran into is that this tick is a carrier or a vector for the protozoal parasite Theileria orientalis subtype Ikeda. And again, we had in this cases where we had the problems in southern Iowa this spring or early summer, this was part of the problem. And so in those herds, we saw some cattle that were anemic. They were very lethargic, maybe, and loss of body weight, etc., with some death loss in those herds. Some herds had pretty high death loss. Some of them didn't have any death loss, and so it kind of varies from herd to herd what that death loss looks like. But last I heard from the diagnostic lab, we had 4 herds that were confirmed positive by PCR for the Theileria orientalis parasite. We did have another herd that sent ticks in that were confirmed to be Asian longhorned ticks. They had multiple dead animals, but they did not send a blood sample in, and so, but we're probably dealing with the similar type area. Most of these herds were in the Van Buren, Davis, maybe Appanoose County, and that tends to be our central spot. I did get a call from a producer in Ringgold County, which is more south central Iowa, and not sure if this was really that or not, but he had mentioned that in May he had a cow that with anemia, diagnosed by his veterinarian, that was non-responsive to oxytet, which would be, they were thinking it was anaplasmosis, but it was non-responsive to oxytet. The the cow died and no other diagnostics were done. So could have been, could not have been, but it's definitely one that we would put up there. But it's too late to go back and try to determine what that was unless we tried to bleed the other cows and see if we could find it in there. But so anyway, at least probably 4 confirmed, 1 suspect, and then another maybe not case that we know of. So the parasite, this protozoal parasite, again, is the tick is a good vector for it. It's the only vector that we have in the U.S. That I know of for it. The parasite itself has been in the U.S. for I believe since the 1990s, sometime in that time, maybe late 1980s, that they first found the parasite, but it's been pretty intermittent because we didn't have a vector to move it around, and it was basically moved around by like other blood-borne parasites, movement from animal to animal by a mechanical vector, like using the same needle, using the same tattoo pliers, etc. That was moved from animal to animal. But now that the tick has been in the U.S. and we have it here, then it's that movement of this parasite is going to be a lot easier now. And the tick can basically pick that parasite up anytime it takes a blood meal from a bovine that is carrying that parasite. It goes through a life stage down in the digestive tract of the vector of the tick, and then it ends up in the saliva. And then when that tick next takes a blood meal, it will inject those sporozoites into the bloodstream. And so you see, we get a lot. Because it goes through that replication in the tick, we get this amplification of the number of those sporozoites versus if we're transferring it needle by needle from cow to cow, we're only transferring 1 or 2 of these of the pyroplasmas within the erythrocytes, which is a pretty inefficient way to move that parasite around versus the tick, because it goes through that life cycle, really gets a lot of those in there. Unlike anaplasm, versus anaplasm, when it gets into an animal, that bacterial parasite goes into a red blood cell, replicates, and then the red blood cell is lysed, and it goes on and goes through to other blood cells. This protozoal parasite actually first infects a white blood cell. And then I've heard some people say that it then goes through a transformation from within that white blood cell and replicates. Other people say it doesn't. This is kind of a generic life cycle for the Theileria for all Theileria organisms. I've heard some people say that orientalis doesn't go through this amplification phase in the white blood cell, but other people say it does, and haven't been able to discern which is which yet. But anyway, once it lyses that white blood cell, then it goes on and as a merocyte and infects the erythrocyte, and then becomes a pyroplasma. At that stage, those will either be will typically be the red blood cells will end up being lysed as they go through the spleen because they're kind of a misshapen erythrocyte, and so we have this non-vascular lysis of the white blood cells. So with what kind of clinical signs we see with this? Anemia, a lot of depression and lethargy, exercise intolerance because of that anemia. Somewhat different than the again from anaplasma, fused to anaplasma, is we really see a lot of the depression and anorexia. These cattle lose weight, and really look pretty rough. Versus anaplasma, we don't tend to see that so much. Some of that is probably associated with the attack on the white blood cells, et cetera. That's leading to some of this. Then we can see jaundice, abortions due to just the hypoxia. If we have enough anemia, then we don't get enough oxygen to that fetus, so we can see some abortions, and then death loss. With the number of ticks we see, when we have thousands of ticks on a single animal, plus we have a protozoal parasite destroying red blood cells, that's why we see the death loss. I've heard some references that the ticks can cause death loss by themselves. That seems like a lot for even 1,000 or several thousand ticks that are to cause enough anemia in a full-size cow. They could probably do it in a calf, but I think probably the accompaniment of the Theileria organism is probably most of our death loss problems.
Yeah, so Grant, there's a question about that that
Yep.
Sandy asked. She said, so cattle death loss is only due to the Theileria and not because of the tick burden. So I think you pretty much answered her question, I think.
Yeah, I mean, there's a report in Ohio 10 years ago of death loss from the tick, but I'm not sure that they looked for the Theileria in that case. But that was a report of 3 mature cows died from the tick. That seems a little far-fetched to me, but I haven't. So I would assume that they probably just didn't look for the Theileria at that time. It's reported high morbidity and low mortality disease. That high morbidity may or may not see a lot of, depending on how it's spreading in the herd. We could see a lot of cattle infected and not much more morbidity, or we could see a lot of more morbidity. It kind of depends on how that parasite is being introduced, how many ticks are carrying it, et cetera. And so sometimes I think we do see the high morbidity. Sometimes if we get a lot of carrier animals, we're not going to see that high morbidity. So that's going to be somewhat variable. Again, typically low mortality. Our initial index case here in Iowa had about 18 to 20 heifers and a bull that died out of roughly 100. So 20% death loss, which is pretty high mortality, which is somewhat unusual for this disease. There were probably some things going on with that herd that probably made that more likely in that case, but it's hard to say. But typically we see fairly low mortality with this. So our risk factors, again, somewhat different than anaplasmosis, is our young animals are some of our most at-risk animals, and so versus in anaplas, we're mostly talking about mature cows being at risk, and our young animals have the ability in their bone marrow to produce enough red blood cells to prevent that. With this disease, we don't see that. Some of that, again, we're probably dealing with the white blood cell loss, and depending on how many of these sporozytes are received by the calf at any time, can really amp up this problem. So young animals are a potential there. Naive animals, and that was one potential factor for this first case in southeastern Iowa. They had a lot of heifers that they bought out of Montana. They'd been on the farm for 4 to 6 months, but had probably never seen Theileria before. The Theileria may have been in this herd already, and the ticks just spread it. But those naive animals may have been at risk versus the native animals may not have been as much risk. So that's one potential. Stressful events, and so bulls in the early breeding season, if we've got a very active breeding season with a bull that's getting run down and gets in contact with this disease, this is probably an issue for them. Fall calving cows, and so this may be a risk for people that are calving here, in particular in our beef herds, starting in August, early September, where we could have some pretty high tick numbers, and those fall calving cows can be at risk, particularly for just stillbirth, but also for, we know that our cows become immune suppressed during calving and have some other issues. So they're at risk. And then if they're not, don't have adequate nutrition or having some other underlying disease in that herd, that's just going to add to that. And so we want to make sure that we're meeting all the other needs of the herd from nutritionally and health, preventive health type matters in order to kind of decrease that risk. So potential icterus, see the fetus there on this cow. This was a case in Virginia, so this was not our southeastern Iowa cow. This was a Virginia case. Liver gets to be enlarged, which we don't typically see with anaplasm. We see the enlarged spleen with anaplasm, but we see this enlarged liver here. A lot of cases with Theileria as well. So control of the tick is going to be important. There's not much we can do about the parasite. There's no treatment for Theileria, unlike anaplasm, where it's responsive to tetracycline. It's a protozoal parasite, so there's no approved drugs that can treat the cattle infected with the parasite. And so it's mostly going to be looking at controlling the tick in order to control whether or not that disease is being spread around. So we've got a couple of options looking at the environment. Like most ticks, they are and one reason why southern Iowa, Mississippi Valley is good is it's kind of a more humid environment. They like that humidity. They don't like to get dried out. And so burning pastures in the springtime can expose any of those larval or other ticks that have overwintered there. And the burning is going to kill them, but then also we don't have that high grass growth that allows those ticks to hide for a while either. So burning pastures could be a way to control that. It may be, if in some cases, it may be beneficial to burn the edges of your pasture if you're worried about ticks moving in. Again, if we can get a couple yard area of control around your pasture, that's going to decrease that. Now, doesn't stop a bird or a white-tailed deer from moving it in, but the ticks aren't going to move past it on their own. So that's one way to control that. Keeping your pastures mowed, keeping that down so that the ticks can't hide. As this day warms up and it gets hot, those ticks are going to go down into the lower in the forage in order to hide. If we can keep those pastures either grazed down or mowed off, we're going to have a little better tick control in those areas. Brush control is going to be important. Again, they like that, with the brushy areas, we get that little micro environment that's a little more humid, particularly cedar trees are known to have pretty high humidity areas. So getting rid of cedar trees may be an advantage, or fencing cattle out of those areas. A lot of times we like that tree areas in order to have places for the cattle to get some shade, etc. But we're going to be having to look at how we can control those as well. So, but brush control or fencing cattle out of the brush is going to be one way to do it. For our animals, we got various ways to get insecticides onto cattle. A pour-on, back rubbers or sprays are also a way to do that. Ear tags are not very effective for tick control. They will keep the ticks out of the ears, but they don't have high enough levels to keep the ticks away from other areas, and so ear tags by themselves are not going to be sufficient for controlling the tick. Pour-ons are not going to be 100% effective, but they do have some control. When we pour an insecticide along the back of an animal, that insecticide moves across the skin, through the oily substance there. And so if we have a petroleum-based product, that helps do that. But it may not get all the way down into that flank areas and on the leg areas. And so you'll have pretty good control on the upper body, probably less control somewhere else. And so, but pour-ons can be effective to some degree, not 100%. So typically your back rubbers and sprayers are going to be our best method to get that insecticide spread over the animal. Permethrins are effective and should do a good job. But organophosphates, which are, or not, I guess from my understanding, aren't available in dairy cattle in general, but in beef cattle, I recommend the organophosphates just because we don't use them very much. And so I'm less worried about resistance having established itself yet. And so when I'm looking at tick issues, I'm probably going to look at an organophosphate as a good insecticide. Our avermectins, the macrocytic lactones, ivermectin, cytectin, etc., also effective as a control method for ticks as well. So my recommended control methods for cattle, would be to pour cattle in the spring prior to grass turnout, typically with a pyrethroid. Just because we don't know what's out there, and I don't want to get too aggressive to begin with unless we know the ticks are out there. So a pour-on is going to help those cattle with flies and ticks and other things as well. So, but then I'm going to want to pay attention to those cows, particularly when we get into a new pasture. We tend to see these ticks bloom. The ticks in these cases in southeastern Iowa, where we had ticks that were out on the pasture having overwintered, we're looking for a host, and then we turn 100 heifers out there and we see that bloom. And so when we move into a new pasture, expect there to be ticks out there waiting for a host to show up. And so you're going to want to check those cows regularly and see whether or not we're having those tick numbers come up. If we start seeing those tick numbers come up, then I would want to look at using a back rubber or oiler or those types of things. Again, pyrethroids may be okay, but for beef cattle, particularly for dairy cattle, but for beef cattle, I would probably switch to an organophosphate at that time when I see those tick numbers coming up. And you're going to, using a carrier of diesel fuel, again, is going to help that insecticide spread over the skin better than other methods. And so using that diesel fuel to carry that, and then keep those back rubbers charged every 2 to 3 weeks, making sure that we've got adequate levels of insecticide in there. Then if we're starting to get excessive tick numbers, then I would consider coming in with one of our macrocytic lactones, cytectin, ivermectin, Dectomax, those types of products, as far as getting those ticks killed on the animals themselves at that time frame, and then maybe consider switching to spraying, and where we're going to go through and spray every animal, with a half to a gallon of diluted insecticide spray at that point, trying to get control. So that kind of gives my overview of ticks and the parasite.
Thank you, Grant. We'll go on with Dr. Brewer then now, and then maybe save questions for the end.
Okay, here we go. So I'm Matt, and I think I'll just sort of reiterate a lot of the things that Dr. Duell had to say. So review is good. Hopefully hear things a couple different times, a few different ways. It'll help you remember. So as Dr. Duell said, we're talking about the Asian Longhorned Tick today. I always do my background and acknowledgments first, just because otherwise I forget them at the end. I did my training at Iowa State, and basically my shtick is I do parasites. So we do a diagnostic service here at the college for all things parasitology, and I teach, and I have a basic research lab. I'm interested in drugs and drug resistance. We have a pretty active trichomonas fetus program. But we do a lot of different worms and arthropods and protozoa. I also have a cow-calf herd in west central Iowa. I've got about 40 cow-calf pairs right now. And my wife is a practitioner. She's got a mixed animal practice, I guess mostly beef, out of Adair. And so I appreciate her doing all that. She's really our herd vet. I'm just sort of relegated to tractors and facilities. She does all the hard work so that I can go and play and be a parasitologist. So I have that luxury. So I have a pretty neat job. Just a plug for us, we do our diagnostic service. You can just send things to the VDL and they'll trickle their way to us. And if you want to get a hold of us, it's fairly easy to remember. If you just email parasite@iastate.edu, that's how you get a hold of us. So we'll just kind of go over some of the things that Dr. Duell had to say. Again, maybe I'll emphasize a few different things, but I'm glad that him and I are in alignment. We didn't arrange that ahead of time, but I think I'm going to tell all the same story, so we shouldn't contradict each other. So we'll talk about the life cycle, talk about Theileria, talk about the interventions, and remind everyone that really we just want people to keep an eye out for these cases of anemic cows and of extreme tick infestation. So just background on ticks, they're obligate blood feeding ectoparasites, so they have to get a blood meal to survive. They can't eat anything else except feeding on a host. They're pretty amazing. They can live for years, potentially without feeding. And so our main concern for ticks is their function as vectors for spreading disease. So they can cause some anemia potentially, but they probably don't kill animals in and of themselves, as we said earlier. And like Dr. Duell said, there's 4 life stages. I'll talk about that a little bit more shortly. And so they have incomplete metamorphosis. So everyone probably knows about, the very hungry caterpillar that turns into a beautiful butterfly. That would be complete metamorphosis. The juveniles don't look anything like the adults, but in ticks, the different life stages are basically different sizes versions of each other. And each stage has to find a different host. So each stage finds a host, they feed, they fall off, they molt, they find another host, they feed, they molt, fall off and molt. They're an adult, they find a new host. And so that's why we call these ticks 3-host ticks. There's a few examples of 1-host ticks, but most of the ticks we deal with are 3-host ticks. And so they're great vectors because they attach very securely to the host. They secrete a little cementum-like substance that allows them to be embedded and stay long-term, feeding for a significant amount of time. They feed on a variety of different hosts. Some are more picky than others. Some are more generalists, and they are very long-lived, as we said. So Hemophysalis longicornis is the important topic, obviously, of today. It's native to Asia and Australia, New Zealand. It's now found in here, I believe, in 21 states. And it's not every day that I see something that I really want to read in the New York Times anymore. But they had this as a story a few years back, even made a story in the New York Times about Asian longhorned tick occurring here. We do have 2 other species of Hemophysalis ticks that look very similar morphologically, but they do tend to stay on their preferred host. So there's a bird one and a rabbit one. So if you look at ticks on rabbits, they often have Hemophysalis, but it is not the same Hemophysalis as the one that we're concerned with here. So Dr. Duell showed this picture. This was just in Drovers last month. I was pretty impressed. It was like 2 days before we had identified the tick, and then like Drovers already had their map updated, so they were pretty timely with that. A little background on Hemophysalis. As I said, they're native to Asia and Australia, so all the little red dots here are where they have been found. This was as of 2020. Okay, so this is historically where they've been found. What's missing from this map is Africa. There's also, I believe, some reports in some of the countries, particularly in the Horn of Africa. So let's just do the life cycle again. So we remember it. The female lays eggs in masses. So these masses usually have several thousand eggs. So I have a tick jar at home. I pick them off my dog. And sometimes if I pick an engorged one off and throw it in the jar and don't have any ethanol in the jar, they'll lay these egg masses and the little larvae hatch. So the little larvae, they're very small. They're maybe the size of like a period on a piece of paper and text. They're very, very small. You rarely ever see them. They have 6 legs instead of 8. They hatch and they feed. Oftentimes, that's on a small mammal like a rodent, but not always. Hemophysalis longicornis in particular is fairly promiscuous in its feeding. Then they fall off, they molt, and then they're a nymph. So then they have 8 legs and they find another host, which often is a small mammal, but doesn't have to be. It can be an ungulate. So they feed and fall off and molt again into an adult. And then those adults most often feed on ungulates such as cattle and deer. And one thing that's important to note is that with tick pathogens, they can either be transmitted transovarially to their offspring or transstadially, meaning through the different life stages. And so for Theileria, there's not evidence, especially for this Theileria, that there is any kind of transovarial transmission. So that means that multiple life stages must feed on the infected animal to transmit Theileria. So that means that either a larva or probably a nymph fed on an animal that was infected with Theileria, then it fell off and molted, then it found another animal that was not infected and fed on it as an adult. Okay, so for the ecology of this to work, it must find an infected animal, feed on it, and then find another animal that is susceptible to the Theileria. And on top here in the upper right, it shows that for most ticks we're familiar with, the female tick is attached to a host and she secretes some pheromones, and the male has to come and find her. Okay, so she just hangs out there and he has to come find her. If you didn't know, male ticks transmit the sperm to the female via their mouth parts, which is a bit different. But so Asian longhorn tick is different. The females do not require a male. To my knowledge, and talking to the guys at NVSL here in Ames, they've only found a single example of a male tick in the United States for Asian longhorn tick. And so they do parthenogenesis, which is pretty interesting. Typically, in most animals, you have meiosis that forms the gametes, the egg and the sperm, and they lack that that meiosis, and basically just through mitosis can form eggs and lay eggs. So they don't require a male, and that's what allows these really massive populations to build up. And as Dr. Duell said, if you have a female hitching a ride on wildlife or on cattle that are on a truck that get trucked across the country, they can drop off, lay a few thousand eggs, and potentially start the life cycle again. So that is important. And so Dr. Duell showed this map. I'm going to show a series of these maps. Different groups have done forecasts for where Asian longhorn tick may establish itself, and Dr. Duell, I think, is correct in expressing some skepticism about how accurate these are. Oh, I'm sorry, I guess I could have had my picture on here. So this is the first one. This is from 2018. This was another one from 2019 that showed a somewhat similar prediction. This was out of a group in 2020 out of Asia. Interestingly, they actually predicted that the middle sort of third of the United States would be the more suitable area. So that was a little bit different prediction. And then this is the most little bit more recent one too. This one came out of K-State, where they made 3 different models, and the dark red is where all the models agreed. And as the color gets lighter, fewer and fewer models agreed on the prediction. And so what I want to emphasize is, if you read these papers, these models are based on mostly 2 things: is where the tick has already been found and what the climate and habitat is in those locations. So they took all the reports of where it's been found so far. They did some measurements of rainfall and habitat and elevation and other things, and they put them into the model. And so these are predictions, they're not boundaries. Okay, so they're based off of places where Asian longhorn tick has been found. It's not necessarily where it's capable or incapable of being established. So I really don't think that this sort of precludes any part of Iowa in particular. I think it's interesting in these predictions, they sort of didn't think that Minnesota and Wisconsin were suitable. But if you look back at the other guys, thought they were suitable. So it kind of depends on what you put in to your model. I really do not think this is going to be relegated to Southeast Iowa, in particular, because cattle go on wheels. I mean, we put them on a truck and we take them across the country. This is some data from USDA. This is the most recent update, but things I think are moving a little slower there in terms of updating this map. So these are ones that are verified by that. So NVSL here in Ames actually verifies all the first county reports. They want to examine these ticks to verify any reports from the field. And so it's been found in a number of different ticks. A lot of them have been found in the environment. Like Dr. Duell mentioned, the one in Michigan was found during surveillance. So people go around and they drag strips of cloth through the grass. It's called tick flagging or tick dragging. And they examine those drags and look at the ticks. Deer are a common host. They've been found on dogs, a variety of small mammals like raccoons and opossums, and then cattle. So a variety of different animals can be hosts. The initial description, my understanding, was a fairly astute veterinarian that observed a really severe infestation in sheep. So not all ticks are suitable vectors for all pathogens. And as Dr. Duell said, really, the Asian longhorn tick is the one tick that is the competent vector for Theileria orientalis in North America. There has been some studies by USDA where they take other types of ticks like dermacenter ticks or dog ticks, also most people call them wood ticks, for example, that are common in Iowa, and they tried to transmit this on cattle and they could not. So the tick has to feed on a competent host. The host animal must be in the transmission site. That tick has to go on to bite a second host that's susceptible. So all those things have to be occurring correctly, or disease transmission will not occur. Okay, so why do we care? Hemophysalis is a potential vector for Theileria orientalis, and Dr. Duell showed us a little bit about the disease and the disease presentation, and that again stems from red blood cell destruction. So after the tick bites, there's an initial phase of the parasite or the Theileria in white blood cells, and then it moves on into the red blood cells. And any of the red blood cells that have this infection or have little aberrations on their surface from being infected get tagged by the immune system to be removed in the spleen. So they either lyse from parasite replication or from being removed via the spleen, and so these animals become anemic. So Theileria is a genus of protozoa, so it's a cousin to malaria, to coccidia, and a lot of these protozoans, we have really poor availability of drugs because they're eukaryotes. They're very much like the host, and we don't have a lot of anti-protozoal drugs. Most of our knowledge of Theileria orientalis is developing in real time. Okay, so in the last 5 to 10 years, we've been seeing this play out. There's not a lot of historical knowledge here, at least in the US, about how this is going to play out. We can extrapolate some lessons from other Theilerias. For example, East Coast fever in the Horn of Africa is caused by Theileria parva, and that's been studied for many decades. And the paradigm there is that movement of animals leads to exposure of new strains and leads to disease. So I was very lucky last spring. I got to take a trip to Kenya to visit some colleagues there, and so this is the International Livestock Research Institute. What they were working on is vaccines for Theileria, but it was very laborious. So in those cases, if you wanted to make a vaccine, you had to go into the location where the disease was occurring, capture ticks that were infected in that location. You see them all pinned to the dish here, so you had to dissect out the salivary glands of those ticks that contain the parasite and basically make an autogenous vaccine for that herd. And so in Kenya, their context was that in particular herds, they might have a flare up of disease, but once immunity develops, things pretty much stabilize. But then if you move cattle into a new area, they see a new serotype, or I'll call it a biotype of the parasite. And if they get that new version that their immune system hasn't seen before, they can break with disease again. And so this is similar to the pattern that we're seeing with Theileria here, even though it's a different species. So Hemophysalis is a vector for Theileria orientalis. There are 3 genotypes. The main one that we are concerned with is Ikeda. That is the one that we have found associated with disease in cattle. There's probably going to be more differentiation of these genotypes. For example, there are Ikeda strains in Asia that are different than the Ikeda strains in the United States, so that is probably subject to develop over time. But for the moment, we're concerned with the Ikeda genotype for Theileria orientalis. And so, as I said, a common pattern that we see with Theileria is that naive animals are most susceptible. This results in sort of a sudden acute disease or possibly none at all, typically followed by periods of stability. So we expect that probably we're going to see some morbidity and potentially mortality associated with this disease. But we expect that once it becomes endemic, we should have relatively stable situation. One thing that I will note is that producers that have cattle die from Theileria, at least for the time being, at least have a little bit of recourse that there's a livestock indemnity program with the federal government that they can get a little bit of payment out of that. I believe it's 75% of the value of the animal. We don't get that for things like anaplasma, and probably as time goes and Theileria becomes ever present and stabilized, that's probably subject to go away, but that's something to keep in mind if you have infected herds. Moving animals into areas with new biotypes then sometimes results in disease, and much different than Kenya, where there's not as much cattle movement. Here we see cattle move across the country all the time, particularly from the east where more of the cow-calf herds are to the areas of the country where grain production is for feeding. Okay, so this was an example of the original case in Virginia. Again, these parasites, the puroplasms, are found in the red blood cells, and they basically look like little basophilic spots. Okay, so it's very difficult, at least to my eye, to tell the difference between Theileria and anaplasma and other things. I had a bull that didn't breed cows a few years ago, and I looked at the blood and I thought it was anaplasma, and I brought it to my buddy here at Iowa State, and he looked at me and said, "Oh, that's just stain precipitant." Huh? So, it's fairly difficult to see these by eye and really know what they are. The PCR at the VDL is very good. I would recommend submitting for that PCR if you have an index of suspicion, or basically if anaplasma is on the list, now probably Theileria should be a differential for that. There's actually several different Babesia and Theileria species which are closely related that infect animals in North America, and they all make these little organisms in red blood cells. And my point is that there's a lot of organisms that make little dots in red cells. And the best thing that you can probably do to get to an answer is to do the PCR. Our molecular section at the VDL does a really good job with this in terms of sensitivity and specificity. And the two main modes of transmission we're currently concerned about for Theileria in cattle in North America are this tick, but also through iatrogenic spread through needles. So there has been some examples of outbreaks in certain areas of the country where cattle had been moved, they were infected with Theileria, the tick was not present, but through sharing of needles and so forth, the disease was spread amongst animals. So this can be spread through needles and so forth. So Asian Longhorned Tick and Theileria orientalis have been in Missouri for years. Now these cases have been found in Iowa. In general, Asian Longhorned Tick is pretty small. The engorged females look like peas. I've seen a few of the cases that have come through Dr. Duell's hands. I've had some producers in Missouri contact me, people that have cattle in Missouri and southern Iowa. This is just one I got in the last 2 days of some pretty severe infestations in southeast Iowa. I'm hoping to receive these today to try to confirm their identity. And there's been a few deaths associated with these. So as you can see, these are really dramatic tick infestations. The last one I got sent from Missouri, I think the practitioner noted he had been practicing in Missouri for 45 years and he's never seen ticks like this on cattle before in terms of the numbers. Again, this is due to their rapid proliferation with those females being able to lay eggs without having a male. And again, a few more pictures. So these are really dramatic tick burdens. If we look at the mouth parts of these ticks, they have this flared palpal segment. You can see by the arrow here, and this is actually quite a beautiful picture. If you can see my cursor, I'm tracing around these two kind of horns. So when they say Hemophysalis longicornis, the longa cornua are the longhorn tick. This is why it's called the longhorn tick. These are the so-called longhorns. So this is actually a very nice picture. So longhorn ticks can be confused with rhipicephalus ticks if you just grossly examine them. So this is a little sidebar, but in terms of babesia and Texas cattle fever that occurs in Mexico and in southern Texas along the Mexico border, we are concerned with rhipicephalus. And actually, well, I don't have, who knows if it's been cut yet, but there are 3 parasites that are line item budget items in Congress. One of them is screwworm that's been in the news and lately. Another one is babesia and rhipicephalus. So we actually have mounted officers that are on horseback that patrol areas along the Texas-Mexico border, and you can see updates on that on the USDA website. The point here is that they have a little flared area of their mouth parts, but it's not the palps. These are the palps of the feeding mouth parts, and this is called the basis capitulum. And you can see these little flared parts, and I'll show some more comparisons of those on the next slide. So rhipicephalus, that's associated with Texas cattle fever, their basis capitulum is a hexagon. It has these little flared mouth part, or excuse me, the base is flared. Whereas if you look at Hemophysalis, the base is rectangular, but the palps that are on either side of this is called the hypostome, that has these little barbs that are like barbs on a fish hook that help the tick implant into the host. I kind of think of them as like, back when everybody had the mutton chop sideburns sticking out on the side. But if you just look at them first glance and you see the little point on the mouth parts out to the side, you can potentially confuse these two. So then just to compare rhipicephalus, which is in the cattle context important along the Texas-Mexico border, they have a basis capitulum that has those points. It's a hexagon. And Hemophysalis, it's relatively square, but their mouth parts are flared. And then in the middle here is dermacenter, that we would call it the dog tick, or many people call it the wood tick. This is probably the most common tick we see in Iowa. And they have a square base, and the mouth parts are relatively square, so they don't have that flare out to the side. So this is one of the features that we look for when we do morphological ID. So if you see ticks on animals and you suspect that they have these pointed base or pointed palps, that would be a reason to send those to us for further examination. So in terms of interventions, we have to think about where are the ticks. Okay, so where are they? They're on hosts and in the environment. So probably most of the ticks are in the environment and fewer of them are on the host. Okay, so some are on hosts such as wildlife and cattle, some are in the environment, and none of these interventions are perfect. Each has a pro and a con. They all have different levels of feasibility in terms of implementing them. So controlling ticks in the environment, as Dr. Duell said, trying to remove tall grass, weeds, brush piles, basically trying to cut down the environment for the ticks that they like, but also for the wildlife. So as I said, a lot of the larval stages like to feed on small mammals. If you can get rid of these brush piles and things like that that can harbor mice and other rodents. Um, Deer are really favorite hosts of these ticks. But as far as I can find, cattle are the main host, or bovines, I should say, for Theileria orientalis. So some guys went and did deer in Virginia after they found that first case in cattle, and they did over 500 deer in Virginia and did not find any Theileria orientalis. So they're probably important for spreading the tick, but not necessarily as a reservoir for the parasite. I give you free range on opossums and raccoons. Ticks and fleas love raccoons and opossums and deer. I think in Iowa, the land use has changed over the last couple hundred years, such that most places are row cropped, but there are still some grasslands and forested lands. But we still have a lot of deer, and ticks love deer. Deer are one of the favorite hosts of ticks for a lot of different kind of ticks. Um, Sidebar is that deer were almost sort of exterminated from the North American continent when people were killing them and taking them back to big cities for human food. Around 1900, there was the Lacey Act. Fun fact, Lacey was a congressman from Iowa, and that act made the wildlife trade illegal. So over the last 100 years or so, we've really had a boom in deer population, and that's true most places in the country, and that's coincided with a boom in the tick populations. I think one thing we have going for us probably in the Corn Belt is that in terms of habitat, if a deer runs across one of these sections that's covered by row crops and a tick falls off and lays eggs, it's probably relatively unlikely that those ticks are going to find another host. So realistically, it's hard to eliminate wildlife and perhaps not desirable. So what else can we do? There's some environmental things that we can do, and these are based on the idea that ticks need moisture. So they crawl up onto the vegetation. They do something called questing, so they hold out their arms to sense their next host. But when they get too hot and dry, they drop back down to the ground where it's cooler and there's more moisture. And that's where most of these tactics come into play. Um, Most of them are not aimed to kill the tick themselves. The exception to that is probably burning. So in some places of the country where burning pastures is common, this is being implemented. This may or may not be desirable. It's more effective in the spring. The caveat to that is that if you burn a pasture and you have these little pockets of unburnt areas, say where there's like cedar trees or there's some little wet pockets, those unburnt areas can harbor ticks. Clearing brush and cedar trees will tend to help. Those cedar trees and brush tend to hold more moisture and they provide habitat for other wildlife. Mowing and bush hogging pastures is somewhat helpful. And again, that's because it decreases the moisture. I think that's perhaps at odds with some people's pasture goals. So that sort of has its pluses and minuses. Folks have talked about spraying the pasture and the environment, which is probably not feasible. And there's off-target effects. And secondly, that only really works in short grass. So if you spray poison into the environment and the grass isn't short, it's probably unlikely to contact the ticks and kill them. So Dr. Duell mentioned some of the different tick control agents. The pyrethrins, if you remember, maybe or maybe not, are from chrysanthemums. So you see them at the farm store, usually in the fall. They come in a bunch of different colors. There's a few places in the world where they farm literally whole farms of chrysanthemums. swath them, collect them, and they crush them, and they extract these chemicals that happen to bind different ion channels in arthropods. The pyrethroids are when the chemical companies took the pyrethrins and they made chemical modifications on them to make them last longer, to make them be more deliverable to animals. But just a reminder that permethrin, which is a type of pyrethroid, is toxic in cats. It's a caticide, kills cats. We have organophosphates used in dip tanks, for example, in Texas and a lot of places in the world. That tactic is less common in the Midwest, but as Dr. Duell said, we can use those in back rubbers and things like that. Avermectins kill ticks, but have a relatively slow speed of kill. It's generally expected that killing ticks within 24 to 48 hours will prevent transmission of disease, but that may or may not totally be accurate. That's just sort of a general rule of thumb. So we have pyrethrins. As Dr. Duell said, ear tags probably can help around the ears, but really aren't going to solve the problem. Back rubbers are probably helpful. Pour-ons, depending on who you talk to, maybe have a more localized effect. If you pour them along the top line, there's probably areas of the animal that don't get as good a coverage. Spraying probably has the best chance to be spread most effectively over the entire body. So this depends on your context. If you have tame cows and you have cows that you can get caught in a confined area, one thing guys do sometimes is take like a garden, like a 2-gallon pump can sprayer. You can spray them and get relatively good coverage. If you got them in pens or run them through the chute, you can get a little bit better coverage, in my opinion. The avermectins kill both endo and ectoparasites. That's why people refer to them as endectocides, because they kill endo and ectoparasites. There are pour-on and injectable forms of them. In terms of ticks, they have a slower speed of kill. There are some preparations that potentially have a longer duration in hosts. There are maybe some trade-offs with that using a lot of avermectins might eventually lead to drug resistance in other parasite populations. So that's probably a consideration here. Organophosphates are an option in sprays or back rubbers. They have a little higher chance of toxicity. But as Dr. Duell recommended, if you're seeing an increase in tick populations, that might be another tool in the toolbox that could be useful. One thing we haven't talked about too much is immunity. So over time, cattle will mount an immune response to ticks. There is a tick vaccine that's available. It's available through veterinarians. It has to be administered fairly regularly, and it's not going to eliminate populations of ticks. The ticks are going to keep biting the host, but it will help decrease the populations. And so if we keep seeing issues with this, that might be a more common tactic. We'll have to kind of see how that plays out. So no intervention is perfect. Some probably conflict with other goals for grazing, nematode control, other goals for wildlife on the farm. But we want to think about both the environment, the need that ticks have for moisture, the ticks that are on the animal, but also realizing that we'll probably have to reapply these, because if we're killing the ticks on the animal, there's more in the environment. They'll find the host again. So we're probably going to have to keep reapplying those ectoparasiticides. So take home points here is that despite the modeling exercises, no place is immune from longhorn tick. My father-in-law always tells me, cattle go on wheels, boy. So, we put cattle on wheels and take them all over the country, and wildlife don't respect boundaries. So I don't think any place is immune. Tick control options are not excellent or foolproof. Each has pros and cons, and so you'll have to do what we can to implement what's reasonable in each situation. Disease may or may not be present, so there's probably some Theileria lurking out there where disease has not occurred. As I said, after initial impact, Theileria is expected to stabilize. Consider testing for Theileria in any case that's similar to anaplasma. And as I said, and maybe didn't emphasize, it's possible to have Theileria and not Asian longhorn tick, and vice versa. I sort of said that twice here, but In those initial studies in Virginia, I believe they went out and crushed up ticks, and about 10 to 15% of the ticks, of the Asian longhorn ticks they crushed up, had Theileria. So it's possible to have longhorn tick and not have Theileria. It's also possible to have cattle that have Theileria but no ticks and move them somewhere else and either have disease or spread it parenterally or iatrogenically. So if you see these suspicious cases, let us know. Talk to Dr. Duell, talk to myself. Please send us ticks if you have these really dramatic cases. For the best results, we would like to see unengorged, non-engorged ticks. They're a lot easier to identify before they get all engorged. If you submit them, prefer to have them sent in 70% ethanol, not formalin, and that's because ethanol allows for better genetic analysis. So if we do want to say crush them up and look for Theileria on PCR, ethanol gives us a better chance to do that. And try not to break off the mouth parts, because as I showed, the tick mouth parts are important for identification, and those can all be submitted through the VDL. If you have any questions, you can send an email to parasite@iastate.edu. So thank you for listening. I appreciate your time.
So the message from the regulatory side, at least in the state of Iowa, is pretty short and sweet. Theileria, not necessarily the Asian longhorn tick, but Theileria is reportable. However, it's not actionable in the state of Iowa. So basically, we don't take any action about it, but we do want to know about the cases. And so I've shared our state vet's number here. Call him, call our AVIC in charge. They communicate both very regularly. If you have a good relationship with your district veterinarian, or your veterinarian has a relationship with the district veterinarian, that's also an option. But we just encourage producers to work with their veterinarians to develop treatment and control plans. But we do want to know about those cases. So that's pretty much the message from the state of Iowa. Pretty short and sweet.
Okay, thank you. So we got some questions here. One comes from Sandy. Was testing done on the survivors in the Iowa herds? And are these herds now considered to be carriers of Theileria? And does that impact the future production? And should the carriers be identified in cold? So it's a question about the status of Theileria once a herd's been diagnosed positive with it.
You want to take that one, Dr. Duell, or I can chime in? I don't know about the survivors. Do you know about the extra testing in that herd?
Yeah, I don't. As far as I know, the only thing that was tested were the samples that were submitted. So we don't have there is a PCR, which is good for identifying particularly cattle that are either dead or morbid. But we don't have a serology yet that we can do surveillance and see what that level is in the herd. And so most of our stuff is going to be looking at acute cases, diagnosing Theileria, and we haven't really moved into the surveillance aspect of that yet. So anything you want to add to that, Matt?
So in terms of them being carriers, it's generally considered that once they have it, they probably have it then, although it may not be at a level that's detectable by PCR. So this is one of those experiments where you hear in vet school, like, oh, in splenectomized calves, you see it reappear. So basically, they get disease, the immune system has a response, it retreats. And this is sort of similar to what we see with anaplasma, that you can have seropositive animals and you can have PCR positive animals. And so they're probably going to be long-term carriers. Those carriers might not necessarily be transmitting the parasite if they have a very low level, if it's not circulating in the blood, if a tick takes a blood meal, if it doesn't have the parasites present. And so if they're PCR negative, I would at least take a guess that those ticks feeding on that animal are probably not very likely to obtain the parasite, but it's probably relatively difficult to identify the carriers and call them. And in some respects, you might want the carriers just in the sense of immunity. So it's thought that actually maintaining a low level of this parasite is what stimulates immunity and protects from future infection. And so that's a feature that we see of a lot of chronic diseases that's beginning to be more appreciated now, like in humans with tuberculosis, for example, that if the pathogen isn't totally eliminated but is maintained at a very low level, that creates an immune response that sort of stops future disease outbreak. And then the next question was, does the parasite affect meat or organs for future consumption? And I do not believe that it affects the meat or organs for consumption. That's not on my radar, Dr. Duell.
No, not yeah. I think it's pretty much blood-borne parasite. So yeah, there's no impact on that I know of. And so yeah, just to follow up what Matt said, from my understanding, again, it goes into really low levels. But then, of course, like during late pregnancy, we're going to expect maybe those levels go back up. And so it's not just going to go completely away in that animal forever. It's going to fluctuate up and down. And so what that transmission looks like is going to vary over the year. And you might get lucky and catch it with a PCR, but until we have a good serology, it's going to be hard to do much for surveillance.
Yeah, I think that's a really good point Grant makes, that stress could cause sort of recrudescence. So say you had some calves that were infected, maybe you didn't have disease, but maybe when you weaned them and moved them somewhere else, you could have some issues, or late pregnancy or any other kind of stress or other disease that's going on could maybe allow it to kind of rear back up. So that's a good point.
All right, then Joellen asked about the 50% mortality number, and Grant put a reference in the chat there if anybody's interested in that, where that came from. All right, so
just to follow up, I think that was a lab study where you basically took the nymphs I think it was the nymphs, and in the lab scenario put them under minus 15 degrees Celsius, so where they got to their cold hardiness level. So but as far as environmental aspects, again, we get any sort of cover they have where they can get insulated in the leaf cover, and those types of things are going to be more protected than that. So that's kind of a 50% reduction was the lab temperature.
all right, and then Robin from iDolls said ticks are sensitive to desiccation, so varying the winter conditions, mortality could be high. So it could be some variation there depending on just how severe the winter is and what the humidity and temperature is like. And then Rick asked, will long range provide adequate ivermectin dose or need to use Poron? So I guess the question is whether the injectable long range will work or whether it should be a Poron.
Grant, you want to take a stab at that one first, and I'll follow up?
I mean, I generally, when not just long range, but any of the injectables, particularly when we're dealing with some of these acute cases, I tend, if we can get to the animals in a squeeze chute and give them an injectable product, whether that's Ivomec or Dectomax or long range, I'm more confident in that than the Poron, because I know we're getting adequate blood levels right away. But as far as I know, long range doesn't have a tick label as far as I know. Matt might want to confirm that. So there's no research done on it. I assume it gets high enough levels for tick control, but I don't know much. So it should. There's the concern about resistance, which we hear with long range all the time. But I'll let Matt's the expert on resistance to that.
Well, you're the guy in the field, so I wanted to hear what the field expert said first. I mean,
Yeah, I mean, I've heard long range is effective, and particularly from some vets in Virginia, kind of use it quite often for control during the summer.
But I think the mode of action or the way it gets into the tick, though, is by the tick eating the blood from the cow.
Yep.
More than directly because you put the poron on the animal's skin.
correct. Yeah, that's correct. It would get into the blood, and they'd have to take a blood meal. So you wouldn't see them like up and die like you would see them if you had pyrethrins or organophosphates. I mean, I think it's one of those things that, like, yeah, it probably works. It's probably an easier thing to do, but it could have long-term repercussions. So the parasitologists as a group are worried about long range and its potential to cause resistance. If you talk to the company guys, their idea is that they say that there's this peak of the drug and then the concentration falls off relatively rapidly, but there is still a tail there in that pharmacological curve. And the concern is that let's say it does work well for ticks, and you use that for a few years, but there's a population of ticks that, as that curve falls off, they're biting the host and they're getting subtherapeutic doses of the ivermectin. That over time that could tend to swing populations of not only ticks but also other endo and ectoparasites towards having a resistant phenotype. That's all kind of being borne out in a real-time laboratory currently. I guess my opinion in these acute cases would be that the pyrethrins and organophosphates are probably a better right now thing, because I think if you give them long range and you have these really dramatic infestations, you're not going to see the real fast results that you might want to see in those cases. It might be I would say that it might be something to consider in herds that are having a problem, but it might not be the best practice as a sort of blanket recommendation across all herds. Is that fair, Grant?
Yeah, and I guess my preference would be I wouldn't reach for long range for tick control. I would typically use it in stalker calves where I want worm control for the grazing season. And if I happen to get some tick control along with it, that's great. But I wouldn't reach for long range first. I would look at the pyrethroids, the organophosphates, and then
One thing to bring up here is that we have a problem with resistance for the worms that sheep and goats get. Almost all the wormers we use for them are becoming less effective, and so the more we use these things, the more likely we're going to have more effects for that later on. So.
I think as kind of that being the sidebar, with sheep and goats where we have homuncus, they're capable of actually killing the sheep and goats. So when we have resistance there, we see it because those sheep and goats get sick and die. With cattle, at least in this part of the country, we tend to not see dramatic, definitely don't really see death from worms. We might see that maybe sort of in the southeast US. Maybe Grant will tell me I'm wrong, but in general, we don't see that dramatic death. And so there's probably a fair amount of nematode resistance, drug resistance in cattle, but we never look for it. The last time we did any kind of national parasite survey in cattle was like 2012 or 2014 or something like that. And speaking firsthand experience, it's a lot cheaper to give animals doses of parasiticides than it is to send me fecal samples for fecal egg count reduction tests. And so that's kind of the decision that people are faced with. So I think that the worry about nematode drug resistance is real, especially in cattle, and we could be losing production dollars on it, but unless we look, we don't really ever see it. But there certainly is that worry, and that's going to keep going over time. And just as a note, there hasn't been a new parasiticide approved by USDA in about 20 years. So ivermectins and avermectins came on the market in the 1980s. Guy that discovered them got a Nobel Prize. It was like a gift from God to have those wonderful drugs. They haven't been around for that long, but in some places they're useless for certain applications. And so I think just long term, we'll probably be hearing more and more about judicious avermectin use. I've even seen some articles in the popular press, like Drovers and things like that, that have talked about judicious anthelmintic use. So I've not to say don't use them, but I think that probably use them in pointed situations, not as a blanket, at least, especially when thinking about long range. It's just my opinion.
All right, we're getting close to the end here. Fred, do we need to be done at 1:30? We did have questions that came in earlier that we wanted to talk about. People asked why we had the parasitologist on to ask about the screwworm that's coming to Iowa or coming to the United States. It's in Mexico, and what's up with that? And should we be concerned or?
So I think we should remain vigilant. I think that the federal government has had a fairly pointed response to that. So just as the background, screwworm was present in the United States for a long time. We developed the sterile fly approach where they irradiate male flies and then drop them into areas, and the flies only breed one time. And then those offspring of, if they breed with one of these irradiated males, those offspring are not viable. And so that tactic was used to push screwworm out of the country in around the 1960s, late 50s, early 60s. And then through cooperation with different Central American governments, they had pushed that all the way back basically to the Panama Canal. But in the last few years, it's somehow gotten behind that line where they were doing airdrops of these irradiated males. And so it's made its way now. My understanding is within a few hundred miles of the border with Mexico. That was a big thing last year. Yeah, there you go. So 50s, 60s. And now it's crept back northward to within a few hundred miles of Texas, and they had reopened the border, but I think they had then recently reclosed it. Dr. Duell might be tapped in more to that, but that from an economic standpoint, watching that border situation for your producers could be a big driver. And when guys want to sell cattle, depending on if that border is open or closed, those flies can feed on live flesh. So most maggots only feed on dead tissue, but these can feed on live flesh and can actually kill animals. So we want to remain vigilant. The government has announced that they're going to reopen and establish some of these fly production facilities, I believe in Texas. Grant, do you want to add to that at all?
Sure. So, yeah, as Matt said, it escaped out of Panama like 4 or 5 years ago. Came, 2023, and rapidly moved up through Central America. In November, it was first identified in Mexico. We closed the border. At that time, it was reopened in February, I believe, late February. We reopened the border. Closed it again in May when it was found in the Veracruz area of Mexico. They'd planned on a staggered reopening starting in Arizona and seeing how that went. But then in July, they found it had moved from Veracruz up to somewhere, I don't forget where that was, but closer to Texas. And so basically the idea of opening the border is if Mexico can control its movement, then we're comfortable opening the border. But so far, Mexico hasn't been able to control that movement. So there tends to be a lot of unregulated cattle trade in Central America and into Mexico, and where cattle are being traded without inspection. And so that's kind of the keystone of whether or not we can open that trade, is whether Mexico can slow that down. As Matt said, we are opening up a fly. 5 years ago, all of our fly control was done in Panama. We've now opened up one in Mexico, trying to control this part of the southern Mexico, and then also reopening one in Texas to have that border stabilized. But if we can control it here, that's a lot less than it is trying to control that whole U.S. border with Mexico. So we really need to get that pushed back down into Mexico, hopefully into Central America, where that flight control is a lot easier to manage.
All right, Fred, time to leave, or do we want to go through a few more questions?
Well, I think we could go. We had a couple of real germane questions that I'd say we've got a question from Glenn. Let's see if we can, put that one out there.
All right. If I can remember back to my days in practice, we used multiple products, say long range and pyrethrin together. So it's a question about using multiple parasiticides.
I think that's reasonable. I think that it depends on the application too. I mean, from a realistic standpoint, most guys only have their cows caught at certain times of the year in certain contexts. So when they see the veterinarian, they're seeing them for nematode control, for fly control. And so I think you have to think about the whole picture. I don't think that you are bound to use just a single product. Grant, do you have anything to add to that?
Yeah, I mean, there's definitely, again, looking at standard parasite control, we would typically a lot of times be using a poron pyrethroid and maybe a injectable avermectin product going out to pasture, so we get worm control and fly control, and so yeah, we typically use those in concert. Again, if I'm looking at an Asian longhorned tick infestation, I'm probably going to look at getting a spray or a poron of a pyrethroid right away, or maybe an organophosphate if I can, and then combining that with an injectable macrocytic lactone, just trying to make sure we get all the tick control we can in those heavy infestation areas. So yeah, you can't really combine a pyrethroid and an organophosphate, but we could probably do the avermectins and a pyrethroid, something like that.
Yeah, and I think especially in situations where, a guy's got disease and he's got these dramatic infestations, he's going to want you to do everything he can do. I see that as totally reasonable. For what it's worth, I use avermectins in calves because I think that's where it has its biggest benefit. I haven't used a avermectin in my cows in 10 years, and I have vet students come to the farm every June or July and take fecals, and they hardly find any parasites at all in my cows. So there's some aspects to host resistance and grazing management. The sheep have kind of caught on to that, the sheep world, that now they're doing more female replacement selection based on worm levels. And there's actually guys now that intentionally give weaned ewes worms, and they see which ones shed a lot of eggs, and those are the ones that hit the road, and they keep the ones that don't shed very many eggs as replacement. So that might be somewhere that we're going. But I think certainly in the context of an outbreak, you're probably going to use most of the tools. I mean, if this is my cows and they look like this and I got cows dying, I'm probably going to hit them with all the hammers. I wouldn't say that at that point I'm not going to couch myself as being worried about resistance, but I am just saying that as a long-term, tell every herd to do it, it may not be as good of a long-term option, but certainly in the context of these severe cases, if it was my cows, I'd hit them with all the hammers. Is that fair, Grant?
Yeah, I think that's I mean, with an infestation, we're not worried about resistance. We're worried about clinical disease, and so, I wouldn't probably, if I didn't know the tick was out there, I wouldn't do long range and a pyrethroid poron going to pasture, just because it's probably not going to be that beneficial. I might do a pyrethroid poron. If I was in southern Iowa, I'd probably do a peri-throw pour on going to pasture, and then monitor their situation and see what happens. And then if it's ticks are coming up, then I'm going to supplement that with something else.
Yeah, I think that's a good recommendation. I mean, going to pasture and worried about ectoparasites, it used to be the general recommendation, say with deworming, was like deworm them and put them on a new pasture. That recommendation is kind of not what we think you should do anymore in terms of worms. So in springtime, those worms, theoretically, a population of those gets killed over the winter. And if you have your cattle on corn stalks and they're going back to grass and you treat them for nematodes and there are resistant nematodes present, then basically all you get in your pasture is a bunch of resistant nematodes. So to me, the most bang for your buck is that midsummer deworming in calves. I mean, that's why we precondition calves and deworm them then, because their worm burdens are relatively low in, say, May compared to like end of June going into July and August. They're going to have a lot higher burden. I think you'd get more bang for your buck treating things for nematodes at that time.
All right, our time is up. I appreciate Dr. Duell, Dr. Brewer, and Dr. Hamilton for joining us. Very interesting talk, and as hopefully you keep us updated as things progress and we get more news on this parasite. Fred, anything else we need to do to close out?
Yep, I'd like to remind everybody that this program has been recorded and will be on the Dairy Team, ISU Extension Dairy Team website. Give us a week or 5 days and it'll be up, and I will send notification that it is in fact archived and available. I'd like to thank everybody for coming to the program, and hopefully we won't have to review this topic again in a short period of time.
Very good. Thanks, Fred.